for coming to my talk. My name is Leonardo Campagna, and I work at the Cornell Lab for Ornithology. And today I wanted to talk about some work I've been doing sequencing the genomes of these 9 seed eaters that we call cappuccino seed eaters, with the objective of trying to understand the genetic basis of the phenotypes that distinguish them. And the rationale behind this is that these are very recent species, and kind of our conceptual understanding of the process of speciation is that eventually things will start at a few key loci in the genome, and only over time, and really in a trajectory that will depend on the system and many different things, we will end up with complete reproductive isolation. So the idea here is that if we can find enough systems that are at this stage, we could start looking at them and looking for those genes and trying to see if we find common patterns. And recent radiations have contributed to our knowledge of which genes perhaps could be promoting speciation. We know that in heliconius butterflies, coloration genes are important. Famous from Darwin's finches, feeding morphology can contribute to this process. And in cichlids, possibly both coloration and feeding morphology contribute to this. So the only way to know if there's a certain category or certain type of genes that particularly contributes to the speciation process is to build our knowledge with additional systems. And the system I work on is known as the cappuccino seed eaters. These are males from the 9 different species in their adult reproductive plumage. These are pretty much the only diagnostic traits that we have. You can see that they vary in these patches, and sometimes you can have a species with a white patch or a black throat patch, and so on in different parts of the body. They also have different songs, so the males differ in their songs, and you can ID a male by its song, and it will have the plumage of that species. Other than that, ecologically, they're very similar. They eat the same things, their beaks are the same. Morphologically, you cannot tell them apart. So, okay, fine. You probably know from the title of my talk and with the picture that I'm showing you that I'm saying I'm going to show you birds that differ in coloration, and guess what? I found coloration genes are different. And so, if you put it that way, it doesn't sound like a big deal, but I think the important part is that other than in the coloration genes, their genomes are extremely different, the same, sorry. So, they really are the same, we'll see. Okay, so they're essentially dimorphic species. These are females and juveniles of the different species. We cannot tell them apart, we cannot identify them, so we don't use them for our studies. And the system has a few properties that make it interesting for a study like this. First of all, these are the ranges of the 9 different species overlaid on top of each other, and you can see that they're mostly sympatric, and there are actually areas here in northeastern Argentina where you can find up to 6 of the species breeding in the same grassland. These are grassland birds. And they have very low genetic differentiation, very low background genetic differentiation. This is a PCA plot derived from about 20,000 SNPs that we got through rad sequencing. And you can see different species are color-coded, and we can pull out mainly only this yellow species here, which is the sister species to the radiation that's up here. Other than that, within the main radiation, there's very little, if any, neutral genetic structure. The other interesting thing in this group is that we have these 8 different species that we cannot distinguish, and they're kind of equally distant from each other. So we could compare species A and B and try to see what genes are different between them, and then do the same in an independent comparison and try to ask, okay, are the same loci popping up again and again? And finally, well, we can date this node, and we know that the radiation must have happened within the last 500,000 to 1 million years. So they're relatively recent. So the data I'm going to show today is whole genome resequencing data. We built a reference genome for one of these species, and then resequenced genomes at a population level at lower coverage. And eventually came up with about 11 million SNPs for these 5 species. I'm also going to show some data where we used long packed bioreads to try to search for structural variants. So if we take those 11 million SNPs, we throw them into PCA. Now we can start seeing that individuals will cluster by species. And this is actually the first time we've ever seen this in this group, and we've been working on it for about 10 years now. And you'll notice that the percent variation that these PC components are explaining is very low. So you can imagine that if you look at this in the Manhattan plot, you're going to get a situation in which only a very small proportion of the gene is involved in divergence peaks. So I'm showing you here two Manhattan plots. Each point here is the average FST value for 25 kb windows. The comparison in the top is, so we have 5 different species, so there's 10 pairwise comparisons. This is the comparison showing the largest number of elevated windows that we call, that we consider are in divergence peaks based on this threshold. Here it's about 0.3% of the genome. These two species here are on the other extreme. It's 10 times less, 0.03% of the genome is above our threshold. All the other comparisons fall in the middle. In particular, this pair here is from 2 species that are allopatric, and this one here, the 2 species, their ranges are mostly overlapping, almost entirely overlapping. So in total, we find, across the 10 different pairwise comparisons, we find about 25 different divergence peaks, and these are rather narrow, and our average is about 250 kb. And in only 5 of these, we find fixed differences. So only 5 of them actually show fixed differences in some comparison. So the next thing we wanted to know is we wanted to focus on one peak and ask what happens across all the different pairwise comparisons. So here I'm zooming into one peak. In this case, they're 5 kb windows, so I'm also zooming in resolution. And I'm going to overlay the 10 different pairwise comparisons. And here you can see that in this case, for this peak, it's actually, this peak appears in 9 out of the 10 pairwise comparisons that I'm showing. So you could ask, okay, what's going on? What's happening could be that each species carries a different haplotype under that region. And one way to investigate that is to take the SNPs that fall under this peak and throw those into a PCA. And if we do that, in this case, we find 3 or maybe 4 clusters of haplotypes that distinguish a certain group of species. So we can do this across peaks, across the different peaks, and I'm just going to show you 3 different ones. This is one I was just showing you. And in this case, this peak, you can see, distinguishes a different group of species, and so on across the 25 different peaks. So the obvious question is, what genes are under these regions? The short answer is that even though there's a very small number of peaks and they're relatively narrow, we find about 250 genes in those areas, so about 10 genes per peak on average. And for about 2/3 of those, we have good annotations and we sort of have an idea of what they do. So the way we wanted to approach this is to ask, is there any specific pathway in which these genes are involved? Is there any specific pathway that's enriched by the genes that we find under divergence peaks? And the answer is yes. The main one is the melanogenesis pathway. So we find genes that are involved in this pathway that control pigmentation, things like the agouti signaling protein, so upstream regulators or downstream enzymes, molecules used in the middle, transporters, and things like that. In total, there's 9 to 10 different genes from the melanogenesis pathway involved in divergence peaks. So let's zoom in a little bit more. So these are the panels that I had shown before, and I'm adding these 3 panels here in which I am going to zoom in and now plot individual FSTs for individual SNPs. And I'm only going to plot those that we consider highly differentiated, so they have a value of FST of 0.85 or larger. And you'll see that, in this case, this is agouti. Most of these highly differentiated SNPs, actually all of them, except one, fall in non-coding areas. They're either upstream or downstream of these genes, but they're never in the coding areas. So that it suggests to us that what's actually happening is that these differences control the regulation of these genes. So these perhaps are enhancers that are controlling the regulation of these different genes that themselves are conserved. And we know that we know from multiple genome alignments of more distantly related species that some of these positions are actually highly conserved and are as conserved as the exons in these genes, suggesting that they are important. So the next thing we wanted to ask is, because there are multiple genes involved in some of these divergence peaks, we wanted to know if some of them could fall within some structural rearrangements. And the way we approach this is to take a second species and obtain long PacBio reads, so about 10 kb on average, and map them to our reference and try to call structural rearrangements. So we did that, we filtered pretty hard, and we ended up with a subset that we can trust. And these are inversions, deletions, and insertions. And you can see that they're mostly smaller, the ones we found, and generally they involve either no genes or up to one, or in one case, 2 genes. And the ones in red are the ones that actually coincide with our divergence peaks. So we find actually only 3 small, I think they're 400 or 500 base pair inversions that coincide with these divergence peaks. But it's not the case that the whole divergence peak is involved in the structural rearrangement, as far as we can tell. The second thing we wanted to ask is, how are these peaks behaving relative to each other? So we want to estimate linkage disequilibrium between the different peaks. In this case, I'm just going to focus on those 5 peaks that showed fixed differences. So those are on these scaffolds. And this matrix is just going to plot the degree of LD between SNPs that are on different scaffolds that actually we know are on different chromosomes. So for this species specifically, we can see pretty high LD. So these SNPs are on different chromosomes that are telling they're behaving exactly the same way. And if we look at another species, we can see that we do find LD between peaks, but it's not the same combination, and so on, as we look at the different species. So we interpret this as possibly SNPs or genes in these different divergence peaks contribute to the phenotype, and there's possibly selection on the phenotype, and that's why these things are maintained in linkage disequilibrium. So to summarize, I've shown you that cappuccinos show differences in actually a very, very small proportion of their genome, but the differences are different in different species. And that many genes that are in the melanogenesis pathway fall within these areas, and that possibly most of these differences could actually have to do with the regulation of the pigmentation genes. So to leave you with some open-ended questions that we have and we can't really answer, we don't know if these are actually the genes that are responsible for prezygotic isolation. It's possible, but it could also be that there are some genes in there that we cannot connect to the phenotype. So that's very hard to tell without being able to manipulate things. And finally, I find it interesting that in birds, it's possible that pigmentation genes actually play a very important role in speciation, because we have an accumulated number of cases in which we have recently diverged bird species that show differences mostly in coloration genes. Okay, so with that, I first wanted to thank our lab at Cornell, the Lovette lab, our funding sources, and I wanted to leave you with this slide here. We have an effort to recruit graduate students to improve diversity at Cornell, so if this is something you're interested, there's a website here that you could find more information. Thank you.